Dr. Glenn, I just want to start with this updated death toll. 212 people now losing their lives. But to put this in perspective, coronavirus still has a survivability rate of something like 98%. So where does this fit in terms of previous outbreaks that we've seen, maybe from Spanish flu all the way back in 1918 to SARS in 2003?
Yes, it's quite concerning. I think it's a little hard to judge at this point what a mortality rate would look like. So that has to be balanced against the level of health care that subjects get, patients receive, et cetera. So I just would say it's a very concerning outbreak. The patients who are infected are very sick. They're in the hospital. The hospitals are overwhelmed. It's really quite a worrisome crisis here.
Well, we did, of course, have the World Health Organization making that emergency declaration earlier today. Yeah, why did it take so long to get to that point, and what does it mean in practical terms from here?
Well, look, I'm not a public health expert in terms of epidemiology. We, as a company, recognize that this is a major problem. I would just point to the fact that it's the flu season in the U.S. looks like it's going to be one of the worst in 10 decades. I just saw the NIAID publish some data saying that there were 8,000 deaths to date, 140,000 hospitalizations. So this happens, on a recurrent basis. In the U.S., we have these novel flu viruses that come through. And really, vaccine, where we work, which is, my interest, is a very appropriate and strong way to respond to these types of problems.
So, Doctor, as you think about developing a vaccine on this, today the CDC confirmed the first person-to-person contraction of the coronavirus. What does that tell you? What information does that give you as you work on a potential vaccine?
Well, I think, we're a vaccine company. We're local, but we think globally. The U.S. is certainly an important priority. But we know that this is transmissible. It doesn't really respect borders. It is, again, I just point, it's very much like what we see with flu every year. We're, as a company, we're accustomed to thinking about how to address this problem. And again, I would say if we could make a vaccine, it would be very, very helpful to control an epidemic and keep people from dying and becoming quite ill. So, the fact that there's transmission is known. It's no surprise. I think we're expecting with travel and some of the cases cropping up that there would be transmission in the U.S. I think the CDC is watching it carefully. One thing that happened, I think, in the past 10 years, as you know, there's been several outbreaks, SARS being one of them. There was a Middle Eastern respiratory syndrome, both coronaviruses. And that's started a lot of people thinking about how to address a crisis like this. So that includes infection control in the hospital, obviously monitoring people who come in from flights from foreign countries, and the development of vaccines and therapeutics that could address these novel viruses.
You're a vaccine specialist, so I'm so curious, how is a vaccine, why would a vaccine be better, be different than an antidote, an antibody?
Well, so that's a really good question. So, they're related, of course. The antibodies that would be scaled up and manufactured originally came from an immune response. The antibodies, I think, are mostly going to be deployed if someone is sick. And that can be quite challenging. It's not necessarily a given that can work, So as we say in our, field, if the cows are out of the barn, it can be very difficult to get them back in. So, depending on how sick, how far into the illness someone would be, antibodies may or may not be helpful, especially for these respiratory diseases. So I think it's something that should be developed. It would be a specific measure. But it's not really clear, whereas vaccines really have a very good track record. I used to be a pediatrician. Vaccines are almost miracles in terms of being able to prevent serious disease. And so I think that's a very desirable outcome if we could develop. And of course, our challenge is timing. Can we develop one in time to be relevant. And I think we can, but that's going to be yet to be seen. And we're working very, very hard to see if we can achieve a good vaccine within the time frame that it could be used.
What do we know about the ability of this virus to mutate, and how does that make it difficult to develop an effective vaccine?
Well, that's a really good question. So it's been a very good time in biotech and vaccine. The technologies are just outstanding in terms of characterizing the kinds of proteins that are on the surface of the virus, how they mutate, what they do, their function, and all that information can be utilized to design an appropriate strategy. So these are coronaviruses. There's a lot of them. Corona, is a spike, so it looks like a crown on an electron micrograph, and the little spikes that stick out are called the spike proteins. They have a function. They usually, what they do is bind to a cell. They inject the genetic material from a virus into a human cell, and then it takes over and makes more viruses. So if you can block that process by blocking the spike protein function, you should have good protection. We have a lot of evidence that that's true with SARS and MERS, these previous viruses, that are quite different. So that's one thing that I wish we had done. I wish we had gotten to a point where we had a SARS vaccine that had been developed. There I would say some promise that if we had a vaccine for SARS, there's enough what we call homology or commonality between the proteins from SARS and this novel Wuhan virus that the immunity could work and could prevent serious illness. So that's going to be a question for the new vaccines. Are they broadly cross-protective? We know we can achieve some of that in flu today. So in the experimental arena, we have vaccines that are in late-stage development that show that they can broadly cross-protect. And so as a virus mutates, the vaccine given last year still has some effect. So that's going to be an important question for the coming year. If we can make a vaccine that's broadly protective, is the virus mutating? Right now we have very little information. We, as a company, were able to get the sequence from the virus on the 9th of January. It was confirmed in a paper and put in GenBank. So the information's all very fresh. But we can compare the genetic sequence of the current virus with SARS, and we can see how much mutation is occurring around that spike protein. So all that information is very useful for us as we design a vaccine that may be useful both now and in the future.